This is a special episode of the Immunology Podcast, Immunology 2023, Day 5. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rad. Welcome back to the Immunology Podcast, where we have conversations with immunologists. Today we're back with our final episode from Immunology 2023 in Washington, D.C. We'll be discussing the highlights of the last day and sharing our thoughts on how it all went. If you're tuning in for the first time, be sure to check out www.immunologypodcast.com or your favorite podcast app to see all of the episodes covering the first 4 days of the meeting. We're going to kick things off in just a minute, but before we get to that,
if you're enjoying this podcast, we invite you to read more interviews on the STEMCELL Technologies website. The Immunology Profile series has immunologists telling their stories, discussing their research, and voicing their thoughts and opinions on current topics in immunology. You can find these interviews at stemcell.com/immunoprofiles.
all right, well, diving right into it then and covering the last few. I got a couple of papers from one of the major symposia looking at mucosal immunity. Both are microbiome papers.
Talks.
And papers, almost papers. Some of them were papers, or multiple papers, and talks. So the first one was Isaac Chiu from Harvard. He looks at nociception and how pain signaling affects the gut. And this time it was a partially a microbiome story, but he really found that in this one that the nociceptor neurons, these pain neurons, are known regulators of gut barrier function immunity, but he's trying to break some of this out. So the first bit is isolate that small bowel pain fibers restrict both M cells and Peyer's patch formation, and that if you get rid of the pain fibers, you have bigger Peyer's patches, which leads to increased invasion of salmonella because they use that as a mechanism on the way through. And then he did some other cool stuff where he went and found that CGRP, which is a nociception peptide, so it's a signaling peptide for nociception of pain, and its receptor RAMP1 on epithelial cells regulates mucus production, specifically in goblet cells. If you knock it out, you have thinner mucus. So then he went and used something we've talked about here before, the DREAD system, which comes from Brian Roth at UNC. These are drug-inducible receptors. So their GPCR is designed for neuronal work, but used otherwise now that are specific receptors that only work to synthetic peptides, and then you insert them in your tissue of choice, and they activate that neuron, those cells that express it, because you do, a Flox-Cree type system. But they're coupled the same ways as a neuronal receptor would be. And so you can actually induce this GPCR, but only with the drug and not endogenously. So you can see what happens at endogenous-like levels, ideally. Sometimes it's overdrive, but you can turn on specific neurons. So they do this to 8 or 9 with receptors to figure out what neurons are important in the study that they're doing. And do this for a couple things, but they really emphasize in this stuff some new work with Tregs. So ROR gamma delta positive Tregs. So these resident microbiome-driven Tregs. And they show that these TRPV1 neurons are important in Treg function. And if you activate the neuron, you have less Tregs, and that exacerbates both DSS colitis and sebrodentium. But there's 2 sets of nerves that can do this, and so they then injected into specific sites antagonists and agonists, and were able to look at vagal nerves versus dorsal root ganglion nerves, and found it's not the vagal nerve, it's a dorsal root ganglion nerve, and that's working through CGRP, not substance P, and that if they do a RAMP1 knockout, it restores the Treg function, and to conditional function as well in the goblet cells. So you have pain nerve leads to downregulation of Tregs leads to worse colitis. So that was cool. The other paper I got to see was by Manuela Rafetelu. She was studying a subpopulation of B cells that are CXCR2 positive and Peyer's patch development, and presented a whole bunch of work that hadn't been presented anywhere before, and was showing that these B cells are needed to reconstitute a Peyer's patch, and that they're important for the patch's response to bacteria. And they also demonstrated that different microbes were important in generating these Peyer's patches. So if you take Jackson mice, they have much smaller Peyer's patches than taconic mice. And there's E. coli from the taconic mice can result in Jackson mice reconstitution of full-size Peyer's patches. They will also find that E. coli Nissl, which is not common in Jackson mice and is considered kind of a probiotic commensal, does this well, but Bacteroides, which are generally considered healthy, good bacteria, don't do this. And they found that there's a known early bloom in Enterobacter that goes away, and it's important for T cell, we think, like like booting up of your T cell program, but they also now think it's important for your B cell program because it's important for recruiting these Pyre patches after birth. And so they do some maternal studies and some tamoxifen-induced systems and show a couple things. One, that it's really important right at birth to get these Pyre patches going as this B cell recruitment. And then secondly, that if you do a tamoxifen-induced deletion, you have diminished Pyre patches after colonization. So if you delete it after they have them, they shrink. And they establish that it's responsible for the trafficking to Pyre patches. And then lastly, they identify that curly, which is known as bacterial amyloid, is responsible for promoting Pyre patch formation. And so that's the protein in the E. coli that's doing it. So those are my 2 talks.
Nice. Very interesting. So from my side, I attended the other symposium. So there were basically 2. So I did the other one. And the first talk was from James Crowe. So he has been a guest in our show like almost 2 years ago. He's at Vanderbilt. And he gave a really, as always, like the same when we had our discussion with him, really cool introduction and examples of the promise of using human monoclonal antibodies for emerging diseases and for therapy, especially for people that are not really responding to vaccination. So basically, he has done amazing work doing kind of rapid response to pathogens and generating monoclonal antibodies with therapeutic potential. And he has this huge system built in which he can get samples very quickly, find from serum, find B cells, find antibodies that are against a particular pathogen, and has a pipeline that allows him to really quickly come up with a handful or even more antibodies against a particular pathogen. And this is very useful. Using monoclonal antibodies can help for pre-exposure prevention. Also, if you have post-exposure prophylaxis, if you know you got infected, you can have them as a treatment. It can also be used. He shows that it's also useful as treatment for mild outpatient disease and also for severe disease. So and this has been particularly important now in the case of COVID, but also in the case of Ebola. And he mentioned one of the efforts of the lab in which they actually obtain very highly neutralizing monoclonal antibodies from a Ebola survivor. And they do this by, they get B cells from the patient, they enrich them based on reactivity to Ebola proteins, and they can either single cell RNA seek the B cell receptors in these cells. But they also have this huge system called Berkeley Lights in which they have like a multi-well, tiny multi-well system in which they can actually look for activation and cytokine production from individual B cells, and then they can kind of go and pick them up individually in this little well, and that makes it a lot easier for them to pick up functional antigen-specific B cells. And so he also mentions examples as other viruses that have been found antibodies against Rift Valley fever virus as well, and they can be extremely neutralizing below 1 nanogram per mil for a neutralizing capacity, which means that a human could be treated with tiny doses of like 1 milligram, which is really small and intramuscular, which really I think it's so promising. Every time I hear James Crowe's presentation, I'm like, oh, this is the future of therapy. It's really cool. So he did give a kind of a lot of, he went a lot on how they went into finding neutralizing antibodies against COVID. And we did discuss that in our episode back then. He got access to very early, like the first samples of COVID patients in the US. And he really used his very established, and I think he has improved it ever since, system to find monoclonal antibodies. He has a lot of assays that allow them to test large numbers of clones at the same time and find the neutralizing ones, and also even titrate them all in as one really large scale system. And what's really cool is that he has a big platform in which they're trying to find neutralizing antibodies against basically all emerging diseases and viruses, which is called the AHEAD 100. So we also discussed it in the podcast when he was in, and I think it's so cool. He's like, every virus, we just want to have like a subset, like a handful of antibodies that are specific against every single potential pathogen out there. And one of the so when we discussed with him back then, one of the very promising products that was coming out of their lab was a collaboration with AstraZeneca, in which they had two monoclonal antibodies that he had identified, and they were really doing clinical testing to see how they would protect against COVID. And so there was also another talk from Mark Esser from AstraZeneca, in which he showed the results of this collaboration and how they use this AZD7442 product to prevent SARS-CoV-2 infection or protect particularly susceptible people with immunological deficiencies, transplant recipients, people on immunosuppressors. And also, he gave an idea of how it was to bring this to the clinic, how they had to reshuffle all the priorities in the company in order to get this out on the market very, very quickly. And so he shows that they test this combination of antibodies, and they showed that it really worked in non-human primates. And he was like, we were very surprised and very happy to see how good they worked in preventing infection. And he went to talk about the clinical trial PREVENT, in which they tested prophylactic injections of vulnerable people. And I think the results were very interesting. So they had people, as I mentioned, immunocompromised in one way or another, or with blood cancer. So people that did not respond usually very well to vaccination. And they showed by injecting a single 300 milligram dose intramuscular. So that's, I think, is very, very reasonable, fairly easy intervention. It could really confer protection against severe and critical COVID up to a year after injection of these patients. And this kind of worked throughout all the initial SARS-CoV-2 variants. Unfortunately, it did kind of stop working by I forgot the XBB strain. So, but it did hold on against the early Omicron variants as well, and it protected susceptible people against the worst of the disease, which I think is very cool because, yeah, we now often forget that these people, we think the vaccines are solving everybody's problem, but that's not really the case. So this is a really good approach, and I hope they're going to continue developing such monoclonal antibody treatments. They mentioned they have a new type of antibody in which they do modifications to extend the half-life and to kind of improve. They are engineering the antibodies to improve their function. And I think that's a lot of research is going to continue in that sense. And another talk that I liked was from Amy Hartman at the University of Pittsburgh. And she was studying Rift Valley virus infection and immunity, as I briefly mentioned it before. So Rift Valley fever virus is a RNA virus, which is mosquito transmitted, but it's mostly a disease of livestock, and it does not transmit between humans. But so Amy Hartman's research showed, and she looked into how those patients that do get infected, what are the effects, because it's usually kind of an underestimated disease. So in cattle, one of the most visible effects of Rift Valley virus infection is the loss of pregnancies by the animals that are pregnant. Those who don't die, if they're pregnant, they usually lose their babies. And so she made a really good point in which it showed that at a closer look at epidemiological studies shows that this is also the case for women that are infected while pregnant, also have a substantial increased risk of miscarriage, and that makes this a very severe, really important health issue for humans as well. And her lab studies the effect of infection in the placenta and how really is the maternal fetus interface, how the virus finds its place there and how it affects the health of the fetus. And she shows that indeed RVFV can infect placenta in the mice. So in rats, she has a rat system in which she shows that there is infection of the placenta of the rats. And then she also makes some experiments using human placenta explants, and again show that this human placenta is susceptible to the infection by this virus. And in fact, this virus replicates more in all placental tissues compared even to Zika virus. So this is a concerning infection in pregnant women. So she has this model, she shows this, and she finds with more detail how the fetuses are being affected. The placenta is increased necrosis and hemorrhage. So that really affects, of course, the fetuses. And she looks into vaccination of dams from this rat model and show that there is this ongoing, there's this vaccination that has been shown to induce neutralizing human antibodies. And she shows that it also could be beneficial to be applied on even pregnant women to protect their fetuses from infection by this virus, because at least in her rat models, it provides protection, particularly through maternal IgG from breast milk. So I think is very important. It's a disease I never really thought about. And yeah, how important it is to keep an eye on everything. And the last thing I want to just shout out, I attended a workshop, the career workshop on how to have a successful postdoctoral experience, which was given by Lori Conlon, who is the director of the Office of Postdoctoral Services and Career Services at NIH. And I thought it was really nice. I just wanted something different. And I wanted to take advantage of this workshop. And she gave a really nice talk about the expectations, about, especially I would say a little bit focused on the American postdoctoral system, which was fine, and also how the NIH, what are the kind of the opportunities the NIH particularly provides. But I want to just say that typical, importance of finding a good mentor. What are mentors looking in a postdoctoral fellow? What is the best way at looking, how to understand your position as a postdoc. I just want to say that, of course, her presentation is hard to summarize, but she did mention a couple of resources that I'm definitely going to check out. So she mentioned that the office from the NIH, the Office of Intramural Training and Education, actually has a YouTube channel, and they have a ton of career-oriented talks in there, fully available, free of charge for anybody. So I would definitely recommend those people that are interested in some career training from the NIH. Seems to be an excellent source. So it's the OITE. If you search for that, Office of Intramural Training and Education from the NIH, you'll find a lot of resources there. And there's also a careers blog from the same office. I think also very interesting. So I like that. I appreciated the AI offering this workshops as well. And that was my day. And this has been our conference.
There you go. It has been. We're done, and you're now a better postdoc.
I hope so. We'll see.
All right. Well, that does bring us to the end of our Immunology 2023 episode series. We've met a lot of people and had a lot of fun here, and we can't wait till next year in Chicago in 24. And don't forget to subscribe to our newsletter at www.immunologypodcast.com. And also find us on Twitter at @ImmunoPodcast or email us at info@immunologypodcast.com with feedback. See you next time.